Okay, in this first video for this module, we are going to discuss, first of all, what gene flow is and why it's important for us to quantify it. So before I give you a definition, I've mentioned a little bit about this in the overview video, but I want you to think first about what you think gene flow is, maybe based on this picture here or based on what you've read in the textbook or learned in different classes. What is gene flow? What do you think gene flow is? And I also want you to think in particular, when you're answering this question, think about how gene flow differs from dispersal and migration. So here in this cartoon, we have some birds on an island, a new bird flying over to them. Does this cartoon illustrate gene flow or dispersal and migration? And what do you think and why? I'm giving that awkward pause in the video for you to think and talk to the computer by yourself. Okay, so in this cartoon, when you Google gene flow cartoon, this is what you come up with. But actually, this doesn't technically illustrate gene flow. It really illustrates dispersal. And the reason for that is because gene flow is the movement of genes between populations. And so what that means is, yes, dispersal and migration are definitely necessary for gene flow to occur, but gene flow only actually happens if that migrant is reproducing and producing baby birds. So that's this picture I have shown here. So in order for gene flow to actually take place, any individuals that are dispersing or migrating into a new population have to actually reproduce. That's the only way that they're actually moving their genes into this new population, is if they're contributing to the new gene pool through reproduction and through their offspring. The next main question to think about as we're framing this topic is, why do we want to quantify gene flow? So we talked about what gene flow is, but why is it important? So in these two hypothetical populations, population A, population B, if we know that gene flow is happening between those two populations, or if we want to figure out if it is happening or not, why is that important for us to know if gene flow is happening and how much of it is happening? What do you think? One of the reasons actually ties into the topic we talked about last time with the single populations module. And that is because populations that have higher levels of gene flow also have higher genetic diversity than populations that are isolated. So if we think about this scenario here in the image where we have butterflies that are separated in some parts by a mountain range, but there's some kind of opening or pass that's allowing these butterflies to disperse across the mountain range. So we're having some, assuming they're reproducing across these different populations, we are having some gene flow that's leading to higher genetic diversity. So if we use the color of those butterflies as a proxy for genetic diversity, we can see that on both sides of the mountain range we have some black butterflies, red butterflies, orange, yellow, etc. So if we have gene flow, we have higher genetic diversity. If we compare that to a situation where we have a continuous mountain range and there's no gene flow, what could end up happening is we now have isolated populations. So again, assume that colors are representative of genetic diversity for these populations. If we don't have gene flow occurring, we can end up with this situation here where now we have isolated populations, no gene flow is happening, so they're losing genetic diversity on both sides of the mountain range because there's no interaction. So as we talked about before, if you have isolated small populations, genetic drift can happen and fix alleles, and so that can happen here in this scenario. So gene flow is one way that can prevent this strong effect of drift from happening and can lead us to higher genetic diversity. Remember, higher genetic diversity means populations are going to be more likely to be able to survive long-term and adapt to any potential changes in their environment. So that's one of the reasons why it's really important for us to know if high gene flow is occurring. And an example of this is in two types of cats that are found on the Texas-Mexico border. And they, in this study here, which is cited at the bottom, I'll put the link in Canvas, they were comparing two different types of cats, ocelots, which are shown here on the left, and bobcats, which are shown here on the right. And in this habitat on the Texas-Mexico border, there's been a lot of anthropogenic influence and a lot of habitat change for these species. For the ocelots, they are habitat specialists, which means that they do not do well in open terrain because of all these human changes. There is a lot more open terrain in their habitat right now. So what's happening is the ocelot populations are becoming isolated. So there's no gene flow between these habitats because they don't like going through that open terrain. And the result is that these isolated populations now have low genetic diversity. Like we just talked about in the last slide, low gene flow, low genetic diversity. In contrast, the bobcats are much wider, they're more of a wider ranging species, and so they are still able to maintain high gene flow among their populations. So the bobcats have been able to maintain relatively high genetic diversity. So this is one example of how knowing about gene flow can help inform us about potential species of conservation concern, and if one species might potentially be able to do better if they're maintaining high gene flow versus another. An interesting caveat to this, though, is that generally we talk about gene flow being a positive thing. So more gene flow, higher genetic diversity. But when we think about gene flow in the context of biological invasions, gene flow can be negative in the sense that it's actually helping to facilitate the success of invasive species. So in these images here, this image on the left is a mosquito fish that's invasive to many areas, including Europe. So all this area in gray here is area where this mosquito fish is invasive. And they did some genetic studies to figure out that within this invasive area for the mosquito fish, there's actually high gene flow among different drainages. And that high gene flow is actually leading to relatively high diversity for this mosquito fish, despite the fact that, as we talked about in the last module, oftentimes with an invasive species, you have a founder effect where they have reduced genetic diversity. But in this case, for this particular mosquito fish, there's actually high gene flow, which is helping to bolster the genetic diversity of this invasive species and is actually helping it be more successful as an invasive species. So I hadn't really thought about that until I read that in your textbook. I thought that was an interesting angle in terms of thinking about gene flow and how it interacts with invasive species. And so in thinking about, so we know gene flow is important. So if we think about how we actually want to quantify that, there are some ways to directly quantify dispersal through technologies like radio or radar tracking. Remember, dispersal does not directly give us estimates of gene flow. We can assume that if individuals are moving between populations, they are probably reproducing, but this method doesn't directly tell us whether gene flow is happening. So dispersal can be quantified directly, and it's not done very frequently because it's very expensive and labor time intensive technique. This picture here, this guy is actually one of my friends from grad school, Josh Stewart. He worked on giant manta rays, and that's one of the things he was doing for his PhD work, was using these techniques to figure out where these manta rays were going. And this pole here is him putting a radar tracker on the back of a manta ray. He got to go to all these cool places like the Maldives, and I picked the wrong topic for grad school. But when he was in these places, they actually had trouble putting some of these radio devices on, and somehow they actually figured out that peanut butter helped to attach the suction cup of the tracker to the back of the manta ray and helped it stick better. That's a not really related to anything, but kind of a fun anecdote. So when we're quantifying gene flow, quantifying dispersal through radio or radar tracking is one way to do it. Like I said, because that's expensive, these trackers are really expensive. It's hard to get them on these animals. So often in molecular ecology studies, we're quantifying gene flow through molecular methods, which is what we'll cover in the next video.